So, hello. Let me do a quick sound check. Yes. Yes, I can hear myself, which is always a good sign that at least the audio seems to be working. Okay, at the beginning, always a few seconds until everything's working. So, hi, here we are again. Hi, and welcome again to another Saturday live stream. I want to thank you again that you have found the time to join me. If you're new to this channel here, well, I like to do a little bit of hands-on microscopy every week. Today I decided to dedicate a few minutes to stereomicroscopy. So, of course, also a little bit of compound microscopy like every week, but I thought, well, why not a little bit of stereomicroscopy as well? And specifically, I've got several insects here, which are basically embedded in resin. Not the spider here. I will talk a little bit about this one more later. But I have a whole box of how do you call this? Insect. I did not make them. Okay, these are commercial, real commercially prepared insects that we're using for education. I'm a teacher in school, and I thought, well, I'm just going to show them to you, and we're going to put a few of them under the microscope as well. Because stereomicroscopy is also sometimes a little bit underrepresented. I just want to do that a little bit today, but I have also prepared a couple of other specimens and samples for the compound microscope. For example, I shaved myself obviously every day because last live stream somebody wanted to see some cross-section of hair, and if you basically put some shavings under the microscope, then you can actually see the cross-section. So there are a couple of things that we can do today. If you have any specific questions or comments or so, then I'll usually adapt the stream a little bit. I'll also tell you a little bit of where I got this one here from. Okay. Usually during the first couple of minutes, it takes some time for people to join in. So basically, hello from Oregon. So I'm saying hello around the world for more people to join in. From New York, from Styria in Austria. Yes, hi, hello. Also back to you from Upper Austria. Yes, off topic for member approvals. I'll talk about this later. Yes, I will do that. I have to, for the form, I have to manually approve you to prevent spam. From Netherlands, from Germany, the United States, University of Florida, Netherlands, okay, and quite nice. If you have any specific comments that are directed to me, then please indicate this by writing at Microbehunter or at Oliver. It's simply easier for me to. Why don't you use at Microbehunter, because then it shows up in a color. And then it's easier for me to actually find it in the chat. Okay, so what are we gonna do today? First of all, I'm going to pick up there where I left off in just a video that I made just a few days ago. Maybe some of you've seen it. I've actually made a video of a so-called a carpet beetle. Okay, so you're able to find that video in this channel. It's just a few days ago I made that video, and I just want to show you the carpet beetle that I found under the microscope. I need to turn on the light a little bit here. Okay, and here it is, and it seems to be sleeping, but usually when I shine light on it, it starts moving around again. And this is an interesting insect. Okay. I've already made 2 videos about this one. I'm a little bit surprised that it was able to survive for 1.5 months in my little Petri dish. You can see the Petri dish up there. And just a few days ago, I was feeding it. I was feeding it some fingernails. So I was scratching off some fingernails, which is a protein called keratin, not carotene. From the carrot, but keratin is written differently. That's the name of the protein of hair and of fingernails, and it eats that because this carpet beetle likes to eat wool. And wool, of course, is the fur of sheep, right? And so that's basically what it eats. And over here, somewhere, I've also found a so-called, I need to maybe zoom out a little bit here. I found, this one over here is the skin of the carpet beetle. It molted because as it grows, it starts to shed its skin. And what I'm going to do now is I'm going to keep on watching it, because this one seems to be the larva stage, and it should actually basically should metamorphose into the adult form sometime. So what did it eat? Well, I have to admit, for over a month, I did not feed it anything. And I thought actually that it's probably gonna die. And then I just checked again last week, and I saw, well, it's still moving around. I couldn't believe it, and that's why I decided to feed it a little bit. And it actually ate up my fingernails. And what I'm gonna do now is, before I actually show you the spider and some of the other insects and the whereabouts and everything, I'm just gonna show you what I have done, and I'm gonna feed it again. But yeah, I'm just gonna do the same thing now that oh, great. I just cut myself. You see, you always have to be careful with sharp blades. Look, this is the thing. Yeah, that I just gonna show this to you. Yeah, this is the blade, and this is the result. Yeah, just you always have to be careful. And of course, what am I supposed to do now? Gee, of course, I'm changing topics again. Yeah, and I have to get myself now some tissue paper. You always have to. You see, that's what I mean with changing. Improvising a little bit, and let's do the following. If you want, we can also look at a little bit of blood later on. But then again, this is something that I've done already in the past. If you want to look at blood, you can make a little blood smear, or what you can do is the following. Just, this is too much blood, I just realized. It's too much. Let's take a little bit away. Let's make a very thin blood film. This is a little bit, I don't know. You see those, I don't know, what did Bob Ross always say? The artist, happy little accidents? Yeah? So, we can have a look at this as well. But actually, let's stay on topic. Actually, it's a pretty deep cut, I just realized. Ooh, okay. I need to... somehow. What I've done is the following. I'm just gonna show you. That's why you always gotta be a little careful. To feed it some fingernails, all you do is you scratch. Yeah. This is a sharp razor blade, which I bought for microtoming. And if you do that, you're actually going to see that there is a little bit of fingernail over here. Yeah. Yeah. So can you remove the tarantula? Here we go. Okay, yeah, but I'll show it to you later in any case, so you might have to click away. Yeah, but honestly, if you're afraid of spiders, I also don't like spiders. Okay, but if you are afraid of spiders, then there is a psychological technique to actually get used to them. You have to decondition yourself. It means you look at pictures of spiders and so on. Yeah, so that's actually a possibility to get your fear, to manage your fear. Yeah, you have to expose yourself to the fear, and then your brain and your body learns to not be afraid anymore. Right? So that's basically what I've done here. So here it is. Let's zoom in a little bit. Okay. Yeah. So I'm gonna see what it's actually gonna feed on it. Right. So that is essentially the first thing. I need to get something, some kind of plaster maybe, but it kind of stopped bleeding already a little bit. Yeah. But that's always dangerous when you're, non-attentive, and then accidents can happen. Yeah. So I'm going to, So that's basically number one. That's a little bit of basically my first step. I'm going to quickly look at a few of the comments here again. I'm surprised that it didn't eat its own skin. Same for me. I also was a little bit surprised. Okay. And I'm just going to have a look. The stream keeps buffering. Is it me or anyone else getting that? Okay. So I don't know if the stream is buffering or not. I know that YouTube changed. You know what I'm going to do is I've got two of them here open, so I'm going to pause it because I've got two windows open, so I hope that it's kind of stabilizing now. Yeah. Okay. So I think the bleeding stopped now. Okay. Yeah. So I hope that this is going to be fine. Yeah. So Yeah, okay, so let's get that sorted out first. The blood we can look at later. What I'm going to show you now, and yeah, and I know that if you're afraid of spiders, then please look away now. I'm going to quickly show you the tarantula. It is only the skin of the tarantula. It's not the full animal. And we've got two of these specimens in our school. And I think, yeah, all of a sudden they were in the display box, and I think that maybe a teacher or a student brought them along. Maybe they were keeping those spiders, and so this is only the skin, right? So it was actually taped close, the lid, and yeah, and if you want to have a better look at it, I'm going to remove now the lid here, and honestly, for me, this is also I've also not looked at a spider of this size under the microscope before, so I just removed the lid, the tape of the lid, just a few minutes before the start of the stream, and yeah, I think it's a fascinating creature, not a, how do you say, one that I like looking at, I have to admit, yeah. But I want to have a look at it in any case under the microscope because, yeah, I've never done this before. Yeah, so that's actually one of the nice examples of stereomicroscopy that essentially, yeah, we can do it like this. I'm going to zoom. We have to zoom out all the way, and it's a hairy creature. Yeah. So, and this is how it looks like. Yeah. And yeah, look at all of this. I don't know. This is a little bit shows that the spider is kind of large. Yeah. But when looking at this, I cannot yet again, despite the fact that it's not one of my favorite animals, I can still nevertheless not avoid the feeling of admiration. Look at because if you just look at the yeah, incredible complexity and yeah, all of those tiny little hair, yeah, I think it's still in its own strange way a beautiful animal. Yeah, so yeah, in that sense it's yeah, quite fascinating. Now the question that I have now is a little bit, and that's something that I would have a little bit also on my list of experiments. That would be, I know that there are some people who are making those resin, on YouTube there are quite a few of those channels where people are putting objects into clear resin. It's epoxy resin, basically just like yeah, this one over here, those insects. This seems to be epoxy resin. And this would be actually an interesting way also to preserve biological specimens. Okay, bleeding luckily stopped. Yeah. And I wonder if it's not possible also to preserve the spider in this way. Yeah. So look at this here. I mean, this is really, yeah. Just considering that many of these here actually probably are also sense organs of the spider. Here now on the back, and here we can actually see this is where it's open, and maybe this is where it kind of emerged. Yeah, on the back part. Yeah, the exoskeleton. Yeah, is kind of is open, and I suppose that this is where the spider came out when it molted. Yeah. So yeah, so it's considering the fact that this is actually an arthropod. Yeah, looks almost like yeah the fur of of a mammal or something. Yeah. Yeah, there is already a comment here from Jesse. My dad used to make the insects in resin. It's a lot of work, and you would want to do a lot of practice before doing a large spider skin like that. I used to help my dad. Yeah, that's interesting, of course, huh? Because just watching some of the YouTube videos where people putting some tiny objects and artworks into resin. Yeah, it does getting everything free of bubbles and so on. That seems to be quite a challenge. Yeah. So yeah, are the these are basically the yeah. Let's have a look. I think pretty much everything is. Where I'm going to go back here. Here. Yeah, look, you have got the 8 legs. I don't want to touch it, you know, because. Yeah. You've got Yeah, here that's the cephalothorax, because spiders, in contrast to insects, have a head and a thorax, which is the chest fused together. That's the abdomen. Yeah, and you've got 1, 2, 3, 4 legs, 5, 6, 7, 8. Yeah, these are pedipalps. Yeah, and the mouth is over here, right? So, wow, it's pretty I think fascinating, but I don't want to have one of these crawling around. Yeah, so this is yeah, just yeah, just careful. Yeah, it's kind of brittle. Yeah. So that is, and over here it seems to be actually broken open, where maybe the spider emerged as it molted. Yeah. So what about the eyes? So, yeah, you're actually asking about the eyes. I don't know. They must be somewhere up here. Maybe these are the eyes here. I don't know. The spiders actually have 8 eyes. So I might have to put this again under the microscope. But I mean, I don't know. They should be somewhere in front here. Let's have a look. And then I would like to show you a couple of the things from. This is the problem, is that the contrast is not quite. These could maybe these are the eyes here. What do you have? This could be, again. I'm not I'm turning on the arrow. Maybe these here. Okay. But I'm not sure about that. Yeah. Yeah, so I mean, of course, no. Of course, when the spider molted, of course, it is taking out the how do you say, the eyes along. But you should see some kind of an impression in the exoskeleton, of course. Yeah, yeah. No compound structure. That's correct. Spiders have lens eyes, in contrast to insects. And what I would like to show you now is also some pictures that Mike sent me just today, a few hours ago. Okay, because these are just also some 3 pictures where we can also see some compound eyes, because we talk about insects and arthropods and spiders. So let's look, picture of an ant. Yeah, so Mike sent them to me. I don't know if you are in the chat as well. Maybe you can also comment on that. And what you have here is, of course, you've got those nice compound eyes, which are made of separate eyes. But the next 2 pictures are also quite nice. This one over here is beetle or bug. Yeah, that is a bug and not a beetle. This is called a stink bug, it's commonly called. And I just want to show you how you actually can distinguish beetles from bugs. Do you see this back part here, right? You see it's kind of transparent. That's basically the lower part of the wing is membranous. So this is, beetles have a full cover. The front wing is all solid all the way back. But bugs, like this one over here, they have the lower part of the wing is transparent and like a membrane. Yeah? That's why they belong to the so-called the Heteroptera. Yeah. And also very nice. Look at this. Yeah, here you can also see the compound eyes quite nicely. Yeah? So thank you very much for the pictures. Okay, and what I want to do now is just still checking a little bit my bleeding here. Just for the fun of it, yeah. As before I show you the other insects, let's quickly. Show you the following. I'm switching over to the scope view. I need to turn this up here. And I'm going to cover the spider again. So I'm going to put the spider away now. So if you are afraid of spiders, you can now join in again. And what you see over here, those structures, I made this a few weeks ago also during a live stream. These are the bristles of the carpet beetle. And I think there seem to be some kind of a defensive structures. And several years ago, I found them individual ones in the dust and had absolutely no idea what they were, until somebody in a German microscopy forum actually told me what they are. They have in German, for those of you who speak German, it's called Pfeilhaare, and because like basically arrow hair. Because they kind of look like arrows, and these are defensive structures of the carpet beetle. And I would like to show you where did I put it? Here it is. maybe I can actually show them to you. So I'm going to put the it back into the stereo microscope here. Here we go. Yeah, it's every time when it's a little bit out of the light, it starts to fall asleep and roll up a little bit. And then when I shine light on it, then it starts moving again. So, but I want to show you now quickly. I need to focus again. Okay. So, and before it starts moving, I now need a, how do you call this? A toothpick. Look, I'm going to now, I hope that it works. The last time it did work, but when you touch the back side, let me move the microphone over. And now I'm going to move a little bit out of the image, out of the picture. I hope you don't mind, but you should still be able to understand me. So I'm going to now, with a toothpick, where is it? I need to hold it maybe a little bit here. Just a second, I don't know where it is now. No, I'm going to zoom out. Because actually last time when I tried that, it actually moved its bristles a little bit. Just carefully touch it, let's see if it, did you see this? Do you see this? Yeah, how it extended? Yeah, the bristles here in the back. Yeah. Yeah, so these bristles here in the back. These are actually those strange looking hair. Yeah, which you do not see the structure quite well under the stereo microscope. Yeah, and maybe after a minute or so, maybe it's going to pull it back again and retract again. And they seem to come off fairly easily. So yeah, they really look like some kind of defensive structures that. Yeah, kind of it's able to shed off. Huh? But I found this kind of fascinating. Yeah, that it responds like this. Yeah, so yeah, I just had this little carpet beetle crawling around on my wall. Yeah, and yeah, and maybe it's going to pull it back again in a minute or so. Yeah? So I'm going to leave the carpet beetle now alone, and maybe maybe here on the back side. Here, you're barely visible. Yeah, these things here, these seem to be individual here, right? And those, yeah, extensions, yeah, like arrows are here on the back. Yeah? So it's a pretty interesting animal, I think. Yeah, but what I heard is it's not a good sign to have them in your home or apartment because they do damage. They do damage textiles and carpets and things like this because that's what they eat. Right, so yeah, so I'm going to leave it now alone again. Maybe it's going to go back to sleep, or it's going to retract its bristles again. I don't know. Yeah, so I'm going to put a little lid on top again, and I'm going to put it out here. So let's do the following. Just for the fun of it, let's have a quick look at my blood. How my blood is doing, because after all, I did not want to have hurt myself for nothing. Here we go. Yeah, here are the red blood cells. Okay. At least I cut myself for, how do you say, the purpose of science. So 60 times. Yeah. But you've already seen them before. They're starting to deform already a little bit because the blood is coagulating. And here it's a little bit better. Yeah. Still liquid. Let's move it like this. Maybe let's remove the color a little bit here. Okay, you get the idea. Yeah. So, but I digress because today's topic should be a little bit more about those insects. I want to show you now again and some more of these insects that are they're all commercially made. And when we got them, I had to think the following: gee, not only do they kind of look fascinating, but I mean, look at those. I mean, they're huge, actually. Some of them are actually wasps and horn, and there actually there's also a description here on the side. Yeah, kind of nice looking. However, of course, under the a stereo microscope, like a scorpion, we can look at the scorpion as well. Under the stereo microscope, it's also possible to look at them. However, of course, because of the surface reflective surface, the image quality is not quite as nice. However, and now this is where it becomes interesting, is this a potential possibility to use this resin also as a mounting medium for microscope slides? I'm thinking, I don't know. I don't know if anyone ever tried it out. But it would have the following advantage, that because many mounting media, when they have a solvent that evaporates. And when a solvent evaporates, then basically it starts to shrink and deform the specimen. But because this is a resin that hardens by mixing two substances, it's an epoxy-based resin, it should not shrink all too much or at all. And I wonder, really, I have no idea, I wonder if it is suitable as a mounting medium as well. Yeah. So this is just something that I'm just an idea that I'm floating, and we're going to geological samples are made. Aha, good. Yeah. Does the label mention? Honestly, I need my glasses now because I have to see what it actually says. Look, oh, I'm going to show you this here. That's the scorpion here. I'm going to. There's a look. The badge produced for Zebra AS in Denmark, made in China. Okay. Wait, what else? Yeah. tstores.co. Yeah. So yeah. That's the question. Yeah? Polarizing filter to avoid reflections. That's interesting. I don't have one right now here with me. But let's put this under the microscope, and I just want to show you a little bit some of the advantages and disadvantages. I'm going to just wipe it clean a little bit, because there are, of course, tiny scratches on the surface, which are now visible as well. So it's now on the stereo microscope. Let's zoom out. So that is the scorpion with light coming from the top. And of course, you see those white scratches, but maybe they disappear if we focus a layer further down. The depth of field is quite large. Yeah, so let's do it like this. Yeah, and you see, here there's scratches, right? And now I'm focusing more on the surface of the animal. Okay. So I don't know. Do I have just a second? It's another question. Let me see if I have some polarizing filter here. Where did I put them? I gotta be careful that I don't hurt myself another time. Here, I mean, I got some linear polarizing filters here. I wonder if this works. Just gonna hold it over the. But normally, we're gonna see. Now the polarizing filter actually causes more dust and dirt. Let's do the following. What happens if I put light from the bottom? This is still from the top. So this is now light from. See, that's. Yeah, okay, that's the stinger. Pretty scary looking as well. Yeah. But because not the insect, scorpion is not an insect, because the scorpion is so dark, you don't see a lot. What happens if I put a polarizing filter on the bottom? Then maybe I'm able to, because the epoxy might also add a little bit of color this way. Yeah, I guess I'd have to experiment around a little more with that. Let's turn off the bottom light and let's turn it on from the top again. So yeah, that's still a little bit better. Just UV cure resin might be easier to work with for such small quantities, no mixing necessary. UV cure resins, that's interesting. Yeah, Heard about that, but never used it. I know that there are those fingernail cure resins available. I tried to use those as well, but they were very thick. They were more like a gel and not sufficiently liquid. Yeah. Yeah. So, but let's have a look at a few others. This one over here. Yeah, it's also quite a nice one. It's going further. I should probably readjust my camera a little bit so that I do not have to refocus all the time. Okay. Yeah, and you have to keep the filter near the camera. Well, in my case, I've got an HDMI camera mounted on the photo tube, and I've got a ring light, yeah, from on the surface. But normally, polarizing filters work if there is some kind of a reflective light. But over here, I think the reason why the image quality is not so good is because you see of those scratches which directly reflect the light, and I think that's a little bit more difficult to remove. Yeah. So, yeah. So you see, it kind of works, but the contrast is not the best. Yeah, as expected. Yeah. So for using this epoxy resin as a way of preserving specimens for stereomicroscopy might have its limitations, and I think we have to somehow make sure that the surface is sufficiently smooth. Yeah. Yeah, okay. Yep. Yes. If you cast, that's yes. I also heard about that. If you have a brittle sample, then you can cast it in epoxy. I've heard that some people are doing actually stone and rock samples this way. They're embedding them first in epoxy resin, and then they're polishing and grinding them down to make them very thin. I've also not done that yet. It's I think a little bit time consuming and requires a lot of patience to do that. Yeah? UV glue, yeah, can be thinner but more expensive. Okay, yeah, I'd have to experiment with that, I think. Yeah, so let me quickly read a couple of other comments here. If they're polarizing filter, okay. Yes, so let's have a look at a few others, and then yeah, there's actually one problem that I also want to show you with older ones. Quickly. Yeah, I think, I mean, I don't know, but the fact that, yeah, look, some of them are actually always repeating a little bit. I think they might have, I assume if you are a company that does that, you're probably going to breed those animals. Yeah? And this one over here seems to be a little bit transparent as well. So let's have a look here. So, yep. Yeah, so I think in order to kind of protect it from scratches, one probably would have to think about actually including maybe a glass plate or something, which is harder than the epoxy on the surface. Yeah. So there is plenty of, yeah, I'm waiting for the summer holidays. I mean, winter holidays are coming up, but I am busy with so many other things, but maybe over the summer holidays or so, I might be experimenting around with those. And just to make sure that there is actually a completely flat surface, I might have to maybe also glue a microscope slide on top. Because, of course, glass is significantly harder and does not scratch. Let's see. Other way around here, yeah, here, yeah, the legs. Yeah. Or another thing, and I don't know if this is actually a possibility. Maybe it's possible to remove those scratches by adding a little bit of oil. No, I don't have oil with me right now, but I do have over here a little bit of glycerin. Yeah, look, I'm just going to show you. That's a little experiment now, okay? Do you remember this? That's the oak wood from last week. And what I'm going to do now is the following. I'm going to try to, it's a little bit greasy, the whole thing. Let's do the following. I'm going to just take a sample from here. And I'm going to put the glycerin on top of the resin. Okay. Let's see if this works. Because see all of those scratches? Let's see if I'm able to make it disappear. Is this gonna work or not? I don't know. Or oil maybe would also be a possible. Nah, you see, it goes into drops. Okay, glycerin at least is to hydrophilic, and it goes into a drop. I'd probably have to get some vegetable oil from the kitchen and try that. Maybe that's a possibility to kind of remove the scratches. Yeah, but glycerin is not a good how do you say liquid for that, for removing the scratches. But yeah, you get the idea. I'm going to wipe it again. Yeah, so clean again. Yeah. I wonder if China has these mega farms to mass produce these study blocks. I think honestly, if you really want to go commercial with these guys, right, then you probably have to mass produce them and breed them, right? I cannot imagine this otherwise. Yeah, just having people trying to go out and collecting them, I don't know. Yeah. Some of them might also be protected, you know, and for this reason, it's I think important. Oh, thank you. This is a surprise. Look what I got here. Some vegetable oil. Yeah, my son is watching the live stream as well, and he just basically heard me say that I need some vegetable oil, and for this reason, I'm going to now try the following. Look, I'm going to do the following now. Can I get some oil? Here we go. Okay. That's nice. So, let's try this again. I'm looking for, here they are, the tips of my pipette. And let's see if vegetable oil is able to do the trick. So here we go again. Let's see if we're able to remove the scratches this way. You see, that's the problem. Is of course it spreads much better. This is of course nice. Look. I think this did the trick. We have to refocus a bit. Let's get the arrow out of the way. Yep. And it's much better. Yep, I think this we can all agree that this actually works much better. Okay. Look, nice mouth parts visible. Yeah, the trunk. Yeah. Okay. Yeah, and that would of course be somehow a completely avoidable if we had maybe a glass plate glued on top. So if this is the case, then maybe we can also try something similar with some other. I'm now going to choose another insect which is not too dark, not an insect. I'm just gonna go for the. I'm now going to go for the, how do you call this, the scorpion. Let's put the scorpion on here. Yep. Yeah. Okay. Also some scratch, not quite as many scratches as the other one, but still. Let's turn it around. What about the other side? Yeah, it's actually better. It's better than the other one, but let's try it in any case. Yep, it also removes those scratches. Good. Yeah, I'm happy that this actually works. Yeah, it works similar to immersion oil. Yes. Well, let's put it this way. It evens out differences in refractive index. Yeah. So because the oil kind of fills those cracks of the scratches, and therefore the light it doesn't bounce around, and yeah, in that sense, it's yeah, makes the quality a little bit better. Yep. Okay. Let's zoom in a bit more. And another thing that I usually do when you take pictures, you're in Photoshop or in GIMP or whatever, you of course you also always want to improve the contrast a little bit. Yeah. Yeah. So I'm just occasionally also reading a little bit the comments here. Yeah. Yeah. Okay, I think you get the idea. And now, last thing, I have to kind of wipe it clean again. So I'm going to do that. And I'm checking the time of 41 minutes because I do have a few other things I want to show you. Okay. But that's, again, as I said before, that's, again, a little bit on my list. On specimen, oh, you don't see what I'm doing concerning specimen preparation is to maybe make these things also at home myself. But whatever specimens you use, they have to be completely dry, I guess, because otherwise, if they're not completely dry and properly preserved, then what's going to happen is you're going to enclose them, and then any bacteria that might still be in there might start to decompose it, and anaerobic bacteria will produce then methane and carbon dioxide as decomposition gases, and then you've got bubbles and all these things. Not so nice. Yeah. So in order to preserve these insects in an epoxy block like this, you have to properly make sure that they're really, I think, completely free of water. Right, and dehydrated. Yeah? And I can imagine that that's actually one of the reasons why they made those, in that sense, maybe those insects are kind of easier to make than maybe other specimens because of their solid exoskeleton. They do not shrink so much, and their shape is quite well preserved. Yeah, but other organisms, yeah, when they lose water, when you dehydrate them, they kind of shrivel up and shrink and deform and so on. Yeah. Okay. Yeah, never put anything wet inside a resin block. They can explode. Ask me how I know. Okay, yeah. Because again, I guess because of the decomposition gases that the bacteria form, right? And then the pressure builds up, and methane gas maybe, yeah, and because it's anaerobic, and then it blows up. As a matter of fact, you have these things not only in here but also in improperly prepared microscope slides. If they're not properly prepared, then what's going to happen is that this can actually blow off and remove the cover glass because the bubbles are expanding, and then this kind of pushes everything apart. So yeah, that's just something that I just want to share with you as well. So I'm going to have to put the oil somewhere now. Yeah, and then I'm going to show you something else here. Because I do have another one, because this one over here, these are modern ones, right? I do have one over here on the side, which seems to be already 40 years old. Let me put this away first. Okay. Let me put this away first. So it goes back into this, and here I'm going to show you this here. It's also a resin block. And this one is pretty old. I know that because there was a label on it. It's when our school was founded. This must have been, I don't know, back in the late 1970s or so. It looks kind of nice. However, there's something interesting here. And I've seen this with several of these. You see actually that there's this whitening. This is where a tape was connected. Yeah. So there is, also over here. So some of them actually will turn white with time. And apparently this is some kind of an oxidation process. Yeah. So this is something that has to be we have to be aware of, and I don't know if it's possible to remove that by polishing it away, right? But that is why am I going into so much detail with that? Because I want to explore indeed those resin blocks as a way of making permanently mounted, how do you say, specimen blocks for microscopy. So here again, stereo. Yeah. Yeah. Again, same issue with the scratches. A butterfly, maybe we're able to see the scales. Have to focus. It's not residue of the tape alone, but somehow the glue of the tape somehow reacted with the plastic or with the resin. Yeah, so it's actually a real decolor. It's not something sticking on top of it, but it's actually a decoloration. Yeah, so there seems to be some kind of a chemical reaction happening with that. Yeah, yeah. So yeah, so here we see this again. It's a little bit low in contrast again because of the reflections. Yeah, but you see at least here the individual scales. Not a very good image quality. I just realized. Yeah. So, could we see the green one again? The green one. The green one. Which one? Were you talking? Marie was asking. I don't know. Were you talking about one of those here? There was actually one, a green one, which no, this one is metallic green. We can have a look at this one here, to see how this actually looks like. Okay. Look, it's a reflection. Interesting. It's a heart. Yeah, do you actually see? Yeah, it looks almost like a heart. That's the ring. How do you say it, from the reflection of I've got a ring light. Yeah, and that's why it looks like this. Yeah. It's this metallic. Yeah, again, turn it around. Wow, look at this, cool. Yeah, so those bright spots, these are indeed the metallic reflections of the lamp. You know what, I'm just going to turn it off. And I'm going to turn on the other one. This one comes a little bit from the side. It's not so good. Yeah, this is now a light from the side. Now we see the scratches even more. I don't know. Yeah. Yeah. So, yeah. In summary, I would probably say if you look, when I look through the actual microscope myself, it looks way better than here on screen right now, because of course the camera also adjusts exposure time and all these things. Yeah. Yeah. And oil water. Yep. So let's have a look here again, and that's the. And one thing that I think I do, how do you say? Let's have a look again here. Okay. What do you notice? What do you notice? No name of insect. What a pity. Just saying. Yeah. Because obviously you need I mean, honestly, I don't know if you breed them, then you should at least know, I guess, what they are. But I consider that a little bit of a pity that there is no more information here available. I don't know if we can get more info by researching the batch number, but I think that's something that has to be actually included all the time. You gotta know what it is, at least some kind of a scientific name. If you already bother making one of these nice specimens, and at least put down the name. But then again, yeah, you'd probably have to know what it is, and maybe the people who made those didn't know that. Yeah. So this is it's a little bit of a pity that I just generally have to say. Yeah. Yeah. I mean, especially as assuming that they were being bred, yeah, in a company. Yeah. Yeah. See, they all have the same. They all have the same sticker on it with the same code, even though these are different insects. Yeah. So that's a little bit of a pity, but yeah, just saying. But then again, I mean, the point of those specimens is not really only identification or science or so, but simply to give kids and interested people kind of connect them a little bit better to nature. I mean, that's the reason why we have those, yeah? Because kids should also appreciate a little bit the beauty of nature this way. So I'm going to move on. I just want to show you something really scary. So it's a topic change. I'm doing a topic change now, okay? If you don't mind. Okay, because I've been talking now about 50 minutes. I'm going to show you the scariest thing that I have, and you're not going to think it's scary. This is scary. You're going to say, what's so special? You know what? It was so scary that I actually even taped it close. And honestly, maybe I'm just going to open it again, and then I'm going to tape it closed again. I taped it closed because it was open before. Let's open so that we see it better. This is actually really something. This stuff, what I have here, killed a lot of people in the past. Nowadays not a problem because we now know what the problem is. But maybe you've heard of so-called St. Anthony's fire before. Not St. Elmo's fire, but St. Anthony's fire. Yeah. This is, in German, you call it Mutterkorn. In English, you call it Ergot. And I cannot open it. It doesn't matter. No, I can open it. Where is this thing? I'm going to do the following. I'm going to use my dangerous razor blade. I'm going to open it here again. No, I can't because it's protected. I have to use a different technique here. And this is really interesting because it is a fungus. You know, it's a wheat grain, obviously. And those black kernels that you see here are not grains, but that's actually the fungus itself that's growing in the wheat. And this produces a very powerful poison called ergot, which causes severe hallucinations. Okay, so this is I don't know who brought this along, but this is we've in the past already retired. We had some of our biology teachers were botanists as well. They were trained botanists, so they had not only teachers' education training, but they were actually pretty much into botany. And from the university, they collected a lot of these samples. And that is an interesting one because if you do not remove those, and back in the day, whole villages died out because of that, right? If the baker, I know that was in the 1950s, there was a famous case in France where almost a whole village, small village actually died. People got, hallucinations and all these things, and because the bread was contaminated with those kernels. Right? Those the ergot. And what I'm going to do, of course, now is I would like to put it under the microscope. Okay, so yeah. And nowadays these things are of course all removed, and everything's checked. But this is a fungus, and this fungus, here it is, looks like this. Yeah. So this basically is the fungus itself. It's not. It looks almost like one of the grains, but actually it is. Apparently the fungal structure itself, and this produces a pretty strong alkaloid, So, and this kind of shows also a little bit that the power of stereomicroscopes. You see, we can simply look at these things a little bit differently just by putting them under the microscope as they are. It looks almost like nothing, but has caused quite a bit of distress. Back in the day. Causes not only hallucinations, but also apparently a constriction of the blood vessels, so that body parts started to kind of fall off and so on. Not a nice poison. Okay. Yeah. So is it like a mushroom? It is a fungus, right? So in that sense, there is of course a connection here. Yeah. So here it's as well. So this is it looks a little bit like one of those grain kernels, but it's not. Yeah. Yeah. It's and they're yeah, significantly larger than those. Yeah. Yeah. So I'm just reading a little bit. Yeah. Okay. No comments here. Yeah. Just wanted to show this to you as well. Yeah. Because I took it along because I actually did want to make a video about this as well, because the next topic that I would quickly like to talk about is the following. Is some organizational stuff. You might, for those of you who have been following the channel now for some time, I've been jumping back and forth. I've got two channels, right? This one that you're watching right now, Microbehunter Microscopy, and the other one is just called Microbehunter, which was the main channel up to a couple of months ago. And what I'm doing right now is I'm changing the format a little bit. So all of the microscopy stuff and hands-on lab work and things, live streams will now be on this channel. But if you're a little bit more interested in more theoretical microbiology, some food biology and so on, food microbiology, infectious diseases and so on, I'm trying to pivot now the other channel that I have, and I'm going to do more talking head explainers. So just in case you're a little bit surprised why there has been this change in format. So all of the microscopy related stuff will continue to be here on this channel here. Yeah. So this is, I'm just reading a little bit here. Yeah. Yeah. So just so that you're a little bit aware of yeah, what's going on. I have to also admit I'm struggling a little bit with my YouTube channels right now, because struggling under quotation marks, because yeah, I'm thinking a little bit of topics, what new things to show to you and what topics to talk about. That's not always easy, because yeah, sometimes yeah, I have to be a little bit creative. It's not always easy to be creative. So. And I'm also checking 57. Okay, you know what? Also something that I showed you in one of my videos very briefly, and I said this, why not? I discovered this here. I discovered this here in my fridge. Toothpaste comparison video. Yes, thank you for reminding me. I gotta get some toothpaste, some interesting toothpaste. This here is, I made a video in my other channel, which I just talked about is it okay to cut away the moldy part and then eat the rest? Isn't this really, I mean, to a certain extent, it does look kind of interesting, right? And I've got to wash my hands a little bit, yeah. And one of the fascinating questions that you're gonna ask yourself, of course, you always wanted to know, why is it that the mold does not grow over here, but grows over here, right? Big question. Well, obviously, because many fruits, vegetables, also the human skin, of course, has a protective layer of cells on the outside, which when intact makes it difficult for fungi and bacteria to grow. And as soon as something is cut all open and damaged, just like my finger over here, right, from the beginning of this live stream, then of course bacteria are able to enter and are able to start reproducing. Yeah. So this is yeah, a little bit just something that I wanted to clarify. So this is really important, that if you want to keep food like vegetables and fruits fresh, you have to really make sure that the outside is remains intact and is not damaged. As soon as there's a little cut or damage, then of course bacteria and also fungal spores can enter and are able to start growing there and damaging it. Oh, okay, thank you. The questions again, but you know what? I want to, because I have not done this, I want to put this also under the microscope. Okay, so that we do a little bit of how do you call this, compound microscopy as well. Just another thing I want to show you where you got to be a little bit careful, and I'm talking a little bit also from my own experience. I like using those tweezers a lot, right? They're super practical and convenient, but they have one huge disadvantage, and the disadvantage is they're really pointed, right? Allows you to pick up small objects, but you can also hurt yourself. Okay, and of course, if you now mess around with stuff like this and then you hurt yourself, you're introducing bacteria. You don't want to do that. So I'm just saying, be careful with this, because things like this happen, right? And that's why some basic hygienic measures are always good. So I just want to look at this because I have not looked at it yet, right? That's the only thing. A little bit of water. A little bit of water here. And I need my glasses. And I don't know, there are so many different, I don't know, probably different fungi over here. There's this gray stuff here, which I assume some fruiting bodies. And there's this one thing, other interesting thing, more or less interesting thing, and this is way too large. If you want to have a safe source of fungi to look at, take some cheese, right? Some camembert cheese, brie cheese. They have got cultures of fungus growing on it, which are defined, not poisonous. But you have to always be aware that whether a fungus produces a certain toxin or not depends very much also on the surface that it's growing on, right? So just because the fungus that grows on cheese does not produce any poisons there, does not mean that it doesn't produce poisons when it grows on another surface. Yeah, because they are able to adapt their biochemistry, and then they will produce different end products, and some of them might be poisonous depending on the the surface that they grow on. But this is way too thick, so I'm going to just remove it again. Still way too thick. Yeah. So next, I press down a little bit here. Not enough water. I'm going to use a pipette now. I need my pipette tips. So, and a short look before I throw it out. You see, that's usually when I see things like this, of course I want to throw them out, I want to discard them, but usually I always have a look at them because I think it's a pity not to throw them out. Yeah? Not to observe them first before I throw them out. Yeah? Way too bright, again. And sometimes I'm able to see some spores. And another thing, a little bit, we are able to see that maybe, I don't know, this whole thing seems to be very hydrophobic. That's why the whole thing is in an air bubble. Yeah. You are able to see the individual cells a little bit, the string-like structures. But honestly, this is pretty much one of the worst. How do you say, specimens. You know what, I'm going to close it a little bit here. Here, this is better. Had the condenser too wide open. Those tiny little dots that you see seem to be the spores. And these are the ones that actually cause the problems because they spread, and when you inhale them, some of them might cause allergies. Yeah. So, and yet more, 60 times. And so just wanted to show this to you as well. And now that we have looked at it, I'm able to throw it out. I'm going to quickly read some of the questions here. A new way to light view objects. I don't want to spam channel names here. You can do that if thank you. Okay. So basically, it's okay to if it's useful. I always say if it is useful for the community. If it's useful for the community, then please share the channel names. Please also include a seed to look at up close. Yes, and this is something that I want to do. Indeed, some years ago, I've actually done a time lapse of seed growth under the microscope. As a matter of fact, the seed roots were growing so quickly that you could actually see them actually grow under the microscope. It's pretty cool. So especially fast-growing plants, so you can actually see that it is actually, you can observe it as they grow. Yeah. Yeah. So I'm going to quickly toothpaste comparison. I'm still looking for some toothpaste that might contain some diatomaceous earth. I have to find one. Huh? So that is basically this was basically the how do you say, the vegetable, the pepper here. Where is it here? Which now that we've looked at it, I can throw away, right? Yeah. The apple seeds, very good. If you want to, I don't know if you, for those of you who have not grown apples yet, what you have to do is you have to put the apple seeds first into the refrigerator for several days. They have to be so-called stratified. That means they have to undergo a cold period of time, and then when you put them out, then they will start to grow better, right? In case you have problems growing apples, just put the seeds into the refrigerator for a few days. So having said that, I'm looking at the time again. The problem that I have with toothpastes is the following. I'm kind of worried if they're very homogeneous, you're probably not going to see a big difference. That's a little bit the worry that I have. That toothpastes being very homogeneous, you won't see a huge difference. I know that in the past they used to have some scrubs that contained, now not allowed anymore. Okay, there's some face peelings or scrubs that contain those rough grains, and sometimes they actually deliberately included microplastics, very tiny plastic grains, now not allowed anymore. In many countries, and now they're including some kind of how do you call this, some of seeds. They're grinding them up, or little wood pieces, and as a scrub, and that would also be interesting to kind of look at. Yeah, so I'm just saying, yeah. Kids flavored toothpaste with the colorings, yeah, but unfortunately we can check, but I'm kind of worried that you will not be able to see a huge difference, no structural things, because if it's homogeneous, then it will look all the same, and there will not be anything interesting to look at unless they added something to it, or unless there is some kind of I don't know, some oil water mixtures, which I think in toothpaste is not, but in face creams they have that. It's an emulsion, yeah. But I'll check. So this is something I just also wanted to show you before I go, because I'm already talking over an hour, because there are a couple of questions still here. And this is the following. I was struggling a little bit with this. Shaving myself, dissolving then the soap and in water, and for whatever reason it didn't. It always starts to float upwards, right? So I have to get a little bit a small sample of this. Why am I doing this? Because last week there was a request to look at the cross section of a hair, and that is very difficult to do, because then you have to embed the hair and then you have to microtome it. But when you shave yourself, if you're lucky, and because I've seen this already, then you might actually get a disc of hair with a top view, and then you're actually able to see that this is the central canal of the hair. I hope that this is gonna work. Okay, no guarantees here, but what I have to do is I have to kind of take some of the sample here, and what I have done is it sticks to the tip. What I have done is I tried to wash it as well. I tried to carefully remove the soap water and so on. And there are lots of skin cells in here as well, I've seen. Yeah, lots of it. Not surprising. Shouldn't surprise us. Look, it doesn't go off easily. Yeah, kind of purifying it to get a nice image would be the next challenge. But for our purposes, I think it's going to be fine. And as you probably already have noticed, my hair is not all black anymore, but I'm also now getting some white hair. And this actually is also quite interesting to look at. And I hope that we're able to find something interesting here. Okay, so yeah. So let me remove this here again. Let's put it into the microscope again. Where is the scope view? It's a little too bright. Oh yeah. Oh yeah. Yeah, so this, for example, is a hair, but it's seen from the side. I just show you that this is going to be a little bit a question of luck of whether we're able to find something. So this here is seen from the side, right? So the hair is kind of extending out like this, and then this is kind of so I hope that I'm able to see find one that's kind of turned up, turned 90 degrees. Yeah. So let's go down here a little bit. Lots of skin. Here is one. Actually, two. Yeah, plenty. Here we go. Okay, so I'm just going to show you. This is actually quite nice. Yeah. So this is basically the request from last week. Are we able to see the hair in cross section? And here, that's it. Yeah. See, it's not even completely round, it's a little bit oval, and here in the center it's darker, so that's the central canal. Down here as well. Yeah? Cross section and the central canal. Yeah. And this is kind of interesting in the sense that if I shave myself, I don't know, once a day in 24 hours, theoretically I can then calculate the average size, of hair growth. So basically, this is what I shaved off from here to here, and see it's kind of a little bit diagonal. So maybe this is approximately the hair growth in 24 hours. Okay. Yeah. So that is the thing, yeah. Where else here? Yeah, there's so much. You see, there's so much other junk and in here. Yeah. Skin cells and soap and so on. Yeah. I don't know if this is the one. Yeah, here that's again. These were again the two that we looked at. Yeah, the cross sections here. Yeah, with the central canal. Yeah. Oh yeah, here again from the side view, the hair, right? And you see also here the central canal here. So you see that this is actually one of the hair of my beard, which is white, and this is still one which is black. A pretty huge difference in pigmentation. Yeah, yeah. So because you're talking about an angle, that this one definitely is cut at an angle. You see over here, it's a little bit wider than over here. Yeah. So it would be now the next interesting thing would now be to look at this the cut edge to see if there's a difference between a fresh razor blade and an old one. I don't know. Is this also one of those here? Maybe it could be with, yeah. So let me go down a little bit with the magnification again so that we get a better overview. Lots of, yeah, and the next thing would of course be to kind of maybe centrifuge it a little bit and purify it. Yeah, here another one, cross section. Yeah, here it is. It's like a disc. Yeah. And that is otherwise impossible to actually get a head-on view. Yeah, because even if you were kind of to glue here vertically somehow, yeah, on a microscope slide, I mean, it would be even a small one. It would be too dark to see. But here we really have a thin, thin slices. Yeah. Yeah. Let me see what some comments there are. Okay. Yeah. Because of all of the LSD comments, gee. Yeah, just saying. I mean, honestly, there are. If you're looking at poisons and toxins, I mean, there is so much of this stuff out there. It's unbelievable. Yeah. And yeah. Yeah. Okay. Here another one. A little bit. We can actually see again the central canal. Yeah. So this is actually something that's quite interesting. A few months ago, last year maybe, I actually did, I think, a live stream where I collected some hair from my students and put them under the microscope as well. Yeah. And then, of course, there are plenty of bubbles here because there's still some soap present. Yeah. So, what time is it? Okay, 15. You know what? For the last 10 minutes or so, there are a couple of questions. Yeah, I'm just going to quickly do this here. Desk view. In case, could you please check for member approvals? Yeah, thanks. Yes, I want to say a little bit. I have to still manually approve each one a person who wants to join the forum. The reason is because unfortunately with AI these days, even the how do you say that there's a question that I'm asking, why do you want to join the forum and answer some questions about the microscope simply to check whether you're actually a real human being. This used to work quite well, but in the meantime with AI is even able to answer those questions. So this is a little bit of a problem. Yeah. Oh, okay. Look, I didn't realize this, that my son also put a plaster here. And so that's a little bit of a problem. So I still have to do this manually. Yeah, insects in resin. It's a lot of work. Okay. I think that's going to be a holiday project for me. Yeah. And maybe a lot of practice. So I'm probably not going to be using real insects at the beginning, and I need to have some molds here. Yeah. And sometimes if you have brittle sample or grainy, you can cast an epoxy resin, but then slice it thin. Yep. This is sometimes, I think, also done with rocks. Is that your school collection? They better not be watching today. Well, some of the students are watching, right? I hope at least. Yeah, because, but the thing is the following, this with the whole, with the watching today, this is probably because of this this fungal poison. But again, honestly, if you fungus and bacteria, many of them produce toxins, and it's kind of really dangerous stuff. Right? That's why you have to be so careful, not making sure that food is not too old and so on, because you can become really ill. Yeah. Could you try wiping the epoxy with alcohol? It might help remove the scratches a bit, maybe. Didn't try it with alcohol now, but I don't think so that it might work, because the epoxy should not be dissoluble in alcohol. And if it were soluble, then I think it would actually make it, how do you say, cloudy, right? For example, acetone sometimes does that. If you have some clear plastic and if you treat it with acetone, it turns white. So this might be a problem, but I don't think that alcohol might work. But I'm going to try it out. Yeah. Never put anything wet inside the resin block. It can explode. Yeah, I think it might be the gas pressure that will form. Curious gets very hot, so the liquid boils, expands, and either cracks, or if you're unlucky, it explodes. Okay, so during the curing process, it can become it's an exothermic process. Didn't know that. This is also important to know a little bit, because maybe this is good in the sense that it might kill off bacteria that might be present. On the other hand, it might damage some of the samples. Yeah. So let me have a look at some of the other questions and comments that we have here. But in the meantime, let's just, because I'm looking at something else. It's because an empty table looks a little bit boring. Okay, so I'm going to quickly look at some of the comments here. Yeah, some trusted staff to help off the workload. You know what I'm going to try to do? And I told myself already many times, and I've not done that yet. I might actually give the community a password, like a secret word with which to register, maybe which I might publish either on the forum or in a video. And then when people type in that code, then they're going to be approved. So they have to watch the video first in order to be approved. So this is kind of, yeah. Did you get my slime mold video? Yes, I think I did get it, but I didn't present it here today. Okay, but thank you. I'll have a look. Maybe the next time, then I'll show it. Okay, but thank you for sending me the video. I got some pictures just today about insects, and that's why I showed those. But slime mold video. Thank you very much. Okay. I did disagree with delegating YouTube and website is total control. Yeah, they're different. views on that. Currently, it's not so much a workload issue alone. It's simply that sometimes I'm getting a lot of emails, also spam mails and so on, and filtering them out can be a little bit of a time-consuming thing. Yeah. So toothpaste is on the agenda. Okay. I'll get some toothpaste, but I'm kind of worried or concerned that it might not be very interesting. Yeah. Seed, toothpaste. Okay. Resin. We talk about resin. Okay. I think I answered all of the other questions. Okay. Polarizing filters. Yes. Okay. Got that. Yeah. Is there anything else I wanted to show you today? Otherwise, I'm just going to keep them for next time. But I think this was pretty much it. I just, before I started the live stream, I was cleaning up my cupboard a little bit, and I found some very old samples still that I might put under the microscope as well. I mean, for example, those here. Yeah, these are also a couple of years old already. These are ants, winged ants. Yeah, that I basically, they were already dead. So I've got a couple of interesting specimens, but this would be, of course, much easier to look at if they were kind of preserved in some kind of a resin block. But then again, yeah, they're easier maybe to manipulate them and hold because they're very fragile. But then again, preparing them and putting them into a resin block might also be a little bit difficult. Yeah, yeah. Electric discharge on a piece of copper tape or something, or like a spot weld under the scope. So basically, if you kind of have an electric spark maybe that makes a trail, maybe in wood. Okay. Yeah. Yeah. No worries if it isn't interesting. It's a question of trying and seeing. Yeah, that's. I also agree with you. Even if it's not very interesting, then we've still learned something. Yeah. And for me, those live streams are actually in case you're wondering, also a way to simply play around a little bit and to look at things that I normally probably would not look at. Yeah. So that's a little bit also one of the reasons why I'm kind of keeping the format of these live streams a little bit open. Yeah. And do some experimentation and lab work on the fly. Yeah. Yeah. So, ladies and gentlemen, I think I'm just going to still call it a day today, because as you probably heard, I've got a little bit of a cold and I'm a little bit sore already. So that's why I've got a sore throat slightly. I'm getting better though. But there is something going around, and like every winter, I like to get a cold a little bit, and for this reason, I think I'm just going to call it a day today. Yeah. So for those of you who basically don't know that yet, I'm doing a live stream almost every Saturday at the same time, which will be always announced a few hours in advance, and then you're of course free to join. And sometimes I decide in a very short time in advance on what I'm actually going to show you. So usually this is something that I'm also not planning ahead a very long time. Okay. I'm going to call it a day, and I wish you all the best. Happy microbe hunting as always. That's what I say. And yeah, do check out the other videos in this channel. And I'd like to also hear you honestly. Maybe that's also important. Do watch some of the new videos that I made in the other channel, in the Microbehunter main channel, which are a little bit more on the talking head explainer type videos, very different from the ones that I'm doing here. I'd like to hear your feedback as well in the comments or in email. Right, because I'm changing the format, or I'm in the process of changing the format of the main channel to go towards more, how do you say, traditional talking head explainers, and I want to do all of the practical fun stuff, so to say, on this channel here. But I'd like to hear your feedback a little bit, okay? Yeah, all the best. Thank you again, and hope to see you next week. Bye bye. The other channel is called Microbehunter. I've just seen there was a comment here, right? So links are in the description.